We are very fortunate today to welcome someone who endlessly loves microbiomes and metagenomes to our seminar series at Penn State Microbiome Center. So Tom Sharpton is an associate professor of microbiology and statistics at Oregon State University, and his science has developed the field for the past decade because it spans the gamut of key questions that you're going to hear about today, and the ones you don't hear about if you're meeting with him. He's got a whole plethora of other topics that he studies because of his plasticity in computational and experimental biological techniques, and a wide-ranging depth of collaboration spanning as many animal model systems as you could possibly imagine. If you look at his CV, he's got buffalo, humans, zebrafish, rats, mice, beavers, et cetera, et cetera, et cetera. So not just satisfied with the evolution of metagenomic changes, he is very interested in the applications of those metagenomic changes to animal health and human health in general. Tom joined Oregon State University as a faculty member in 2013 after obtaining a Ph.D. in microbiology from the University of Sunny California at Berkeley and postdoctoral fellowships at the Gladstone Institutes. He has since developed an internationally recognized research program spanning some of those computational tools, databases. I think there were 7 or 8 that I counted on his CV, as well as his animal microbiome reductionist experiments that he produces from his lab. This amounts to about over 75 manuscripts, editing and advising for a variety of microbiology journals, including several from the ASM, so Tom's a good person to know if you're submitting your papers to M Systems or Applied Environmental Microbiology. Trainees, hang out with him. Maybe you can send him some of your papers. And he serves as the founding director of the OSU Microbiome Initiative, which has been quite successful as a leading initiative organization across the country. Tom is freshly back on the post-COVID give talk routine. Academic life is back. We're lucky to have his first week at doing that. I think he was just over in Woods Hole, Massachusetts. Took a short drive over to here before he heads back home. As a part of it, our acknowledgement, thank you for visiting the center. We want to present you with this glass plaque from the Penn State Microbiome Center. So you might put it somewhere special. It looks like that right there. Awesome. Let's give Tom a round of applause and look forward to hearing it.
Well, thank you, Seth, for that lovely introduction and for having me out here. This is, as Seth noted, the first trip that I've taken since COVID started. I took my last trip literally weeks before we went into lockdown. And so it's like coming out of a fog. And it's been so much fun to sit down and meet with people and be reinvigorated and excited about all the different research that I'm hearing from with different folks. So I'm really happy to be here. It's been a great time so far, and I look forward to those of you I haven't had the opportunity to meet with yet, the opportunity to sit down and chat some more about your science. What I'm going to talk about today is a theme that emerges out of the work that we've been conducting in our research program, but we're not the only people who are exploring this research theme. In fact, one of the beautiful things about microbiome science is it's a broad and diverse community, and there are a lot of people exploring complementary questions to one another. Today, I'm going to talk about some of the work that we have done that has advanced a particular hypothesis that many different groups are exploring. And I think there's strong potential as we continue to move this hypothesis forward in our evaluation of it to fundamentally transform how we think about medicine, health, and even life. And so I hope that you have a good time going across this journey of different vignettes over the next hour with me. Now, to just provide some background, we're very familiar with a pandemic at this point, right? We're living one. It's moved into an epidemic stage, but we have very acutely become aware of the importance of understanding the distribution of disease across the planet. Well, I'm here to tell you that that's not actually the first pandemic that you have experienced over the course of your life. We are living through a chronic disease pandemic. Chronic diseases account for the biggest medical burden in our society. We're talking about things like type 2 diabetes, inflammatory bowel diseases, neurodegenerative and neurological disorders. And these are among the leading causes of death in our country. Oftentimes, we just go through life kind of accepting that they exist. But I suppose I kind of wonder if at the end of it all, that's really something we need to accept. Our chronic disease is something that we can prevent. Can we improve the diagnosis and management, and ultimately the treatment of these different types of disorders? This is obviously something that many different people care about deeply. But despite the extensive amount of research that has been conducted in this area, frankly, we struggle often to explain the origins of these diseases. We know they manifest. We don't really understand why or how. Well, one interesting observation that has led to a paradigm in how we think about chronic diseases is the fact that their incidence has sharply risen over the last 50 years, right? It's across chronic disease categories. And it is timed with the rise of industrialization across the global economy. In fact, if you look at historical epidemiological data, what you'll find is that there's been a transformation in the global distribution of all sorts of chronic diseases. Here you see body mass index, which is indicative of, or an indicator of obesity. But basically every single chronic disease is undergoing a similar temporal trajectory, where we start off back in the 70s in cooler colors across most of the globe, warmer colors in areas that have been subject to industrialization. And as market economies have come online, as even by 2014, we've seen the entire map redden. Body mass index has been increasing globally over time. This has raised a really interesting question. Has industrialization caused things to change in our environments that have contributed to the rise of chronic diseases? There are genetic precursors of disease. We know this, but genotype ain't changing rapidly enough to account for those transformations over time. This strongly suggests an environmental component to the rise of chronic disease. Thank you very much. And so if we think about what is correlated with industrialization, well, there's a lot of change. We benefit from a lot of that change. I mean, just look at this amazing facility that is around us. The fact that I'm able to fly here and see all of you wonderful people. We have industrialization to thank for that. But consistent with that process are changes in our food, the pollutants we're exposed to, the drugs that we consume, and as is increasingly common, with as many of us still live in a pretty isolated environment, the social experience that we manifest. Well, these are massive changes if we think about the lifestyles that we experienced prior to industrialization. Now, the reason I'm introducing all of this to you today is because despite looking at these different components and the relationship with different types of chronic diseases, there still remains a lot of information about the emergence of these diseases that we haven't been able to explain well. And I and others tend to think that that's because we've been looking at a limited component of the system of health. And that given recent innovations and discoveries in microbiome science, perhaps we need to be more carefully considering the role that the microbiome plays in our health and how these transformations that have emerged over the process of industrialization have impacted our microbiomes. Now, I talk about microbiomes, and what I really mean is the gut microbiome, because that's where I focus, but there's no reason to be exclusive about that fact. Different microbiomes across the entire body are probably playing a role in physiology in related ways. But we know a lot about the gut microbiome. Again, this is the collection of microorganisms that reside in our gastrointestinal tract, and they interface with all sorts of aspects of our physiology. They can do things like modulating our nutrition, mediating our immune state and protecting us from infection. They can detoxify and even toxify certain types of environmental chemicals we consume. And provocatively and amazingly, they can impact our behavior and our cognition. These are all aspects of physiology that are intertwined with different types of chronic diseases and disorders that we experience. And so these observations have raised a lot of interest in understanding what the microbiome's role may be in chronic diseases. What I think is particularly interesting about this is that these observations suggest that our physiology comes to depend upon our microbiomes. It's a component of our homeostasis. It's functioning in a way that is benefiting or contributing to how our body operates. And some of this, I think, makes sense if we look at this through the lens of evolutionary biology. Because you see, our ancestors didn't exist in a microbial-free world, right? They were colonized with microbiomes as well. They came to probably depend upon, or at least be influenced by, the functioning of our gut microbes. And so when you see some sort of deer walking around in the wild, well, how much of its fitness in nature is impacted by the particular assemblage of microorganisms that reside on its body? Well, this is interesting to me to consider because what it suggests is possibly just possibly, over evolutionary timescales, our ancestors evolved in a way that came to depend upon the functioning of their microbiomes. We don't know that for sure yet. I'll lay out some evidence today that suggests maybe that's true. All right, but what the reason this all ties into chronic disease is because if we arrived as a species with a physiology that depends upon particular microbiome functions to be executed to maintain our homeostasis, and if industrialization arrived and impairs that functioning by changing our microbiomes, then possibly the reason we experience certain chronic diseases is because we've done things to our lifestyles and to our environments that affects how our microbiome functions and erodes our physiology, resulting in a breakdown in it and a manifestation of chronic diseases or disorder. So that's the background. That's the principal hypothesis that we're entertaining in our lab. We're interested in understanding, to be more explicit, how do changes in our diets, the drugs that we consume, and the pollutants we experience impact our microbiomes to result in cascading consequences on our physiology? And in particular, do these changes to the parts of our microbiome that we may have come to become physiologically dependent upon drive these different types of diseases and disorders? To explore these questions, my research group applies what we call a systems biology approach. And this is just a fancy way of saying that we make a lot of different measurements about a lot of different features, and we use computational and statistical tools to analyze the data. So what we think about is this interaction chart right here. We think about this a lot. We are curious about how measurements of host genotype, measurements of the microbiome, and this might be 16S data, metagenomics, metatranscriptomics, what have you, and measurements of our environment, maybe metabolomics data, maybe it's geographic information, maybe it's how much chlorophyll's in the grass that a particular wildlife species is consuming. Okay. How they all interact with one another to explain variation in host phenotype. That's the systems biology approach. There's a lot of computational and statistical modeling involved. Ideally, once we identify interactions, we go into an experimental model and prove cause and effect. That usually proves harder than it sounds, but that is the goal. So this is the approach that we tend to leverage. And today I'm going to talk to you about how we use this approach to resolve specific questions that relate to this overarching hypothesis that I've outlined today. I'm going to take you on a little bit of a whirlwind journey through 3 different areas of research in our lab. I'll tell you about one story where we seek to define the functional connections between the microbiome and host health. I'll then tell you a little bit about how we're developing high throughput tools to rapidly measure how different types of environmental exposures impact the microbiome. And then finally, I'll very briefly discuss some of the microbiome's link to vertebrate evolution. I think this aspect of research is really fascinating, but probably, at least in our lab, the most nascent. So let's begin by zooming into the work that we're doing to understand how the functional capacity of the gut microbiome links to health. Now, the way that we tend to go about conducting this sort of an approach is to step away from the traditional 16S data. Normally, I would take a step and explain what that is, but I'm just going to make the bold assumption that everyone in this room is aware of what 16S data is. I think that's fair to do. And I just want to get everyone on the same page briefly about the alternative approach that we use in this regard. This is known as metagenomics, shotgun metagenomic sequencing. So the idea here is metagenomic sequencing affords us with information about not just who comprises a microbial community, but what those organisms are capable of doing, at least based upon the genes encoded in their genomes. The way that we generate these data is we take a community of microorganisms, right? And we extract all the DNA. We're going to shotgun sequence this DNA. So we're going to chop it up into tiny little bits, and we're going to sequence all these little fragments. Those fragments are randomly distributed across the length of a genome. These blue lines represent the fragments, the length of the genome for the myriad genomes present in the community. Since some of those fragments come from taxonomically diagnostic loci, we can get information on who is there. But since other fragments come from protein coding sequences, we can annotate those fragments and we can get a sense of what genes are encoded in the microbiome. In other words, we can profile what these microbiomes are capable of doing at the bulk level. It's not perfect, but it gives us some insight. All right, so we're not the first people to do metagenomic sequencing by far. This has been around for a while, and we largely have Craig Venter's epic trip around the world on his yacht to thank for the advent of this technology. And it has been applied to the study of chronic disease. But what's interesting is most of the research that has been using metagenomics to study the microbiome's role in chronic disease has focused principally on this context of a single disease. So they'll look at people with type 2 diabetes, and they'll look at people that don't have type 2 diabetes, and they'll ask, well, what are the gene families that differentiate diseased individuals with type 2 diabetes from those that don't have it? Fascinating, we've learned a lot. But there might be something about integrating all that data together to determine whether or not there are some genes that are disease specific, or if there are other genes that tend to explain a variety of different diseases. Maybe those types of microbiome genes represent common pathways that the microbiome is using to interface with and possibly contribute to physiology. That was our goal in this analysis. And so what we did is we took, at the time, all the publicly available human gut metagenomic data that had been produced. This represented metagenomic sequences from across 8 different clinical studies of the microbiome. And we integrated them into a statistical framework. And this allowed us to identify gene families that were encoded in the microbiome that were disease specific versus those that were disease common. The way that we did this, just very briefly, is we took this metagenomic data from healthy individuals as well, or I should say, diseased individuals as well as their paired controls from these different studies. We obtained the publicly available metagenomic data from these investigations, and we compared those data to an extensive genome database of microbial sequences. In so doing, we can basically determine which protein families or gene families are encoded in these metagenomic samples, and we can determine the relative abundance of each of those genes in every single sample. Regression approaches were then used to identify protein families that differentiate diseased and healthy individuals respectively for each of the different studies. So what do we learn when we do this? Well, the first chart that I'm going to show you here, I show simply because we all love looking at ordinations in microbiome data, right? But I don't actually personally think it's all that exciting to look at. We're going to find some more exciting results in just a minute. But yes.
Is the control group, is that grouped together as a lump sum from all of the studies, or are they individually assessed per disease?
Individually assessed per disease, but there are some diseases that are represented in multiple studies. And so we then group those studies, control samples together and model out study effects. Does that make sense? Yeah, perfect. So when we look at the beta diversity of the protein families that are encoded in these metagenomes, these are the results that we observe. We are not looking at taxonomic information. The beta diversity here is built the same way you would be doing this with taxonomic information, but it's based on the profile of KEGG orthology groups that are encoded in these metagenomes, for those of you who are interested in such technical details. What you see here is in red are the cases. These are the diseased individuals, and blue are the the control individuals. All right, asterisks mean that there are significant differences between the cases and the controls. However, if you look at this data, you'll see that, well, the effect size aren't really that big. Yeah, there's a difference, and it's not just dispersion that explains this difference. But the effect sizes aren't really big. There doesn't appear in most chronic diseases to be a fundamental restructuring of the microbiome's functional capacity, with maybe, for example, the exception of Crohn's disease. Okay, but that could be in part because all microbial organisms tend to have housekeeping genes. And so maybe we shouldn't expect there to be a fundamental restructuring of the microbiome's functional capacity. What we did instead, and I think is far more interesting, is we asked what are the specific gene families that stratify cases and controls for each of these different studies. And we call these disease indicators. This is just a fancy way of saying it's a protein family that's more or less abundant in diseased individuals and their respective controls. Now, what we're going to do is we're going to build a chord diagram together today. And every single arc, a component of this arc, every color of this arc represents a different diseased group of individuals, represents a different disease, and the length of the arc corresponds to the number of disease indicators that we identified for that particular disease. So here's type 2 diabetes, and it has a reasonably large number of disease indicators compared to UC ulcerative colitis, very small number of disease indicators. So you can see that some diseases like liver cirrhosis and Crohn's disease have a lot of protein families that discriminate their microbiomes from their respective controls compared to other types of diseases. That's interesting. But what's more interesting is when we ask if any given indicator for a disease is shared with any of these other diseases. And sure enough, this is what we observe. The arcs represent indicators that are common to 2 or more diseases. All right, so this protein family here is an indicator of Crohn's disease and obesity and type 2 diabetes and liver cirrhosis. Now, if you do the statistics on this, it turns out an indicator of one disease is more likely than you expect by chance to be an indicator of multiple diseases. Disease is defined by a change in protein pathways that are commonly perturbed in chronic disease individuals, much greater than we expect by chance. So here's an example of a couple of specific protein families to help put the rubber on the road. You see a situation here where lipopolysaccharide biosynthesis, which is a pro-inflammatory compound, is increased in individuals who have colorectal cancer, liver cirrhosis, and Crohn's disease, all inflammatory diseases. I'm not saying that the microbiome is causing these diseases, right? And I'm not saying that LPS is the mechanism that it uses to do so, but I ain't that a tempting hypothesis to go and explore. What we actually think is going on is these diseases are causing changes in the microbiome that results in the expression of gene families that modulate severity of phenotype. But we have a lot of work to do to prove that out. This differs from other situations where we have this, disease-specific indicators like glycosaminoglycan glycan degradation pathways. GAGs are carbohydrates that line the mucosa of your gut. And we know from prior mechanistic studies that gut microbes that can consume these GAGs make the intestinal cavity more, the epithelial tissue more sensitive to inflammatory attacks and aggregates disease severity. Here we see that these GAGs are enriched uniquely in Crohn's disease afflicted individuals, which is actually kind of surprising to us. We thought it'd be a more common pathway across multiple disease types. So by conducting these types of studies, we can get information about protein families like lipopolysaccharide biosynthesis that may be involved in, that might have to be appropriately optimized in their functioning of the gut to maintain health, as well as protein families that may underlie some of the etiology around disease or its severity in individuals. So just to summarize, we can conduct these types of integrative metagenomic investigations to improve our understanding of the potential pathways through which microbiomes may interface with our physiology to improve our understanding of the context of the microbiome's relationship with disease. It takes us a step beyond just understanding who is present and allows us to conduct, to apply a more holistic perspective about the types of pathways the microbiome may execute to contribute to our physiology in a health-related context. Ultimately, we're hopeful that we can use these indicators to improve our diagnosis of specific diseases and advance the discovery of different types of novel therapeutics and preventative strategies. Let's move to the second narrative. I'm realizing there's no clock in this room, so just give me 1 second. All right, about 30 minutes. Thank you very much. And let's talk a little bit about an area where we have a lot of active investigation at the moment. And this is where we are developing high throughput tools for screening how different exposures impacts the microbiome. Now remember, the principal objective that we have with respect to this hypothesis is not just understanding how the microbiome relates to physiology, but determining how our changing environment perturbs that relationship. So here we seek to determine how different types of exposures in our environment affects our microbiome and whether or not there are compensatory health effects as a result of that. So one of the big challenges that we have with this area of research is the combinatorics problem. One of the most personalized components of your existence is your exposome, the exposures that you experience on a daily basis. What foods did you eat today? What kind of water did you bathe yourself in, assuming that you even took a bath, right? What pharmaceuticals are you taking? These are all very personalized, and you're putting them into your body, or you're covering your body with them, and that's affecting the chemicals that your microbiome is experiencing. That chemical ecology defines the context of the types of organisms that live in your gut largely. So how on earth do we zero in on specific exposures that impact the microbiome? Well, there are a variety of different resources that people have come up with, but toxicology has largely leaned on the zebrafish model system for conducting these types of investigations. There's a lot of benefits to using zebrafish in exposure science. One is that it's ridiculously cheap to rear these organisms, which means you can rear a lot of organisms on the same budget you would normally do a mouse study for. Large numbers of organisms means many different types of exposures. Large numbers of individuals per exposure if you think variation is going to be a pain in the rear. Okay. They also grow real fast. And I'm not going to talk about it today, but what this means is that you can actually do transgenerational impacts of exposure. And there's some really exciting things coming out in that area with respect to the microbiome. And then we also have a lot of automated exposure and phenotyping tools. All right. And so, like I said, toxicology has really begun to lean heavily on zebrafish across the entire globe as a resource, a research resource for these types of problems. Well, as it turns out, the zebrafish is also a great model for studying host microbe interactions. And it's probably the workhorse model that we've used in recent years in our lab for this purpose. There are a lot of interesting features from a microbiome experimentation perspective with zebrafish. With larval fish, we can easily and very inexpensively rear germ-free fish. Very easily. It's trivial. You just bathe the embryos in a couple of different chemicals for a little while and you're good to go. So that means that we can do all sorts of manipulations to our fish at high volume and determine microbiomic effects pretty trivially. But only for larval fish. We can't get them to grow into adulthood germ-free. They don't survive. They develop a massive intestinal colitis. That's a whole other conversation that we can have. But we also have a lot of automated phenotyping tools, and you're going to learn about one today, right? And we've been doing a lot of work in zebrafish to build it out in collaboration with others in the field, particularly Karen Gilliman, Tamara Tall, John Rawls, and others, to really develop it as a useful foundational research system from host microbiome interactions. Just to put in front of you a couple of highlights, we showed just a year ago that the zebrafish gut microbiome actually has a conserved phylogenetic core, irrespective of where in the world you appear to grow the fish, which strongly suggests that the fish is selecting for particular microbes within its gut. Whether or not those microbes are themselves having a beneficial effect on physiology has to be determined. We also have looked at zebrafish metagenomes, and we've been able to find that the functional capacity of the zebrafish metagenome correlates pretty well with that of humans. Not as well as what you see in mice. Here, every single dot is a KEGG orthology group. You can see there's a pretty decent correlation here. This correlation is significantly greater than what you would expect if you looked at free living communities, by the way. All right, so that suggests that there is some translational utility to zebrafish when we're thinking about the microbe's functional capacity as with respect to what you'd see in humans. It's worth noting, however, the kinds of taxa that live in the zebrafish gut are fundamentally, generally speaking, distinct from those we see in humans. So it's an important caveat. Well, using this system, we've been able to advance a couple of interesting stories around exposure science impacts on the microbiome. For example, we showed a couple years ago, this is our first entry into the field, that exposure to the drug triglycerin, this is a consumer-grade antibiotic that you probably put on your body or may even accidentally consume because it's in all sorts of consumer items like toothpaste, hand soaps, et cetera. It perturbs the composition of the microbiome in zebrafish. Working with our colleague Mike Kent, we've been able to show that exposure to a whipworm, Pseudocapillaria tomentosa, links to variation in the gut microbiome. And if you look at the amount of intestinal inflammation, the change in the microbiome's composition explains how the amount of damage that the worm does in the gut. But correlation doesn't equal causation. Inflammation could very easily be having an exacerbating impact on the microbiome. That's something we're currently following up on and looking for chemicals the microbiome may be secreting to modulate immune status or the worm's infection burden in the gut. And we've even used metagenomics to show that modulating a single dietary component, low zinc, relative to the otherwise identical diet, has a subtle but significant change in the microbiome's functional capacity. We've done all that work in adult zebrafish where it's difficult to prove cause and effect. So the thing I really want to spend some time talking to you about today is what we've been able to do in larval fish, where we can zero in better on the microbiome's functional role in physiology because we have access to germ-free animals. So in exposure science, embryonic zebrafish are commonly subjected to high throughput screening assays where we're interested in determining how a particular chemical perturbs the developmental physiology of the fish. So these embryos are frequently deposited in 96 well plates, and columns on the plates are subject to different exposures, maybe different chemicals, different concentrations, so on and so forth. The larval fish grows out, and then we put the plates, so the larval fish is growing in this well. We take this plate and we put it into something called a ZMOD machine. And this is just a fancy device where there's a camera that sits over the plate. We flash light in a dark light cycling, and we measure the amount of movement of the fish in the well during the dark and the light periods as a measure of behavioral activity. A fish that moves a lot is hyperactive, could be anxious. A fish that moves very slowly is hypoactive, and maybe lethargy or depression would be a particular behavioral phenotype if you wanted to get translational with it, but we usually don't apply such terms. So we thought, well, could we integrate microbiome research into this system and transform the scale at which we're conducting these types of investigations? So I want to just very briefly introduce to you a model toxicant that we're studying. This is called benzoapyrene. This is a polycyclic aromatic hydrocarbon. You are almost certainly exposed to it today, unfortunately. It's a byproduct of combustion. It's in your food, it's in your water. Sorry, it sucks, it's there. It does give you cancer, and it also causes a variety of other physiological aberrations. And increasingly, we're realizing that it causes neurological and behavioral aberrations. But those aberrations are quite varied, and we don't understand why. So in fish, zebrafish, we know that if you expose embryonic zebrafish to benzoapyrene, the fish develop neurodevelopmental aberrations. Okay, they become relatively hyperactive. And if you look epidemiologically in children who are exposed prenatally to benzoapyrene, there's a statistical enrichment for them to have ADHD. So there may be some relationship here that's worth sussing out. But again, it's an extremely varied relationship. So we wondered, well, maybe the microbiome's playing a role here, but let's just ask this broad question without any preconceived bias. What happens to the microbiome in fish that are exposed to benzoapyrene? Does the microbiome in such fish change in a way that's linked to behavior? So we took a boatload of embryos, 550 different fish, and we deposited them across these 96 well plates and exposed them to 0, 1, 5, or 10 micromolars of benzoapyrene. And then we phenotyped their behavior, and then we dissected their intestines. This is one of the reasons it's oftentimes great to be the boss. I was on the one sitting under the microdissection scope trying to do this, but I have a great team of people who did this on every single one of these fish. And we did 16S profiling on our microbiomes. So what did we find? Well, if you look at this is taxonomic data, all 16S. If you look at the data diversity of the microbiome as a function of benzoapyrene exposure, green is 0 and warmer colors are greater concentrations of benzoapyrene, you see an association between benzoapyrene exposure concentration and the composition of the microbiome. But it's extremely varied. It's extremely varied. Like the variance explained while significant is only 1.25%. Turns out microbiome assembly, especially in fish, is a messy stochastic process. And what we think is happening is that the benzoapyrene exposure is putting a slight direction on how that assembly occurs. It's meaningful in the sense that it's significant, but we don't know yet if it really has a strong effect on the outcomes of the behavior of the fish, but there does appear to be a connection. We also looked at the microbiome's relationship with behavior in these fish, and we do see that the amount of activity of a fish can be significantly explained, again with low variation, by the composition of the microbiome that has assembled in these fish. Okay, cooler colors represent lower levels of activity than warmer colors. So what we're gleaning here is that benzoapyrene exposure is impacting how the microbiome assembles, and that assembly is correlated with the behavior of the fish. But at the end of the day, what we really want to know is if these things are any sort of meaningful causal relationship. So enter the germ-free derivation model. We took the germ-free derivation approach, and we took these embryos and reared them in a way where they're never exposed to any microorganisms and subjected them to the same exposures as compared to embryos that were conventionally reared. We then measured the behavior of these fish, and we asked the simple question, does the presence of the microbiome or the absence of the microbiome affect how benzoapyrene is impacting the behavior of the fish in these two different groups? What do we find? Let's first look at what happens with no benzoapyrene at all. Okay. Here, what we're seeing on the y-axis is a total amount of movement by the fish when the light is turned off. And you should note that when the light's on, the fish that kind of slows down, right? It's a little anxious and shy, right? It's a little scared. It's got a fear response. And so you see most of its movement in the dark cycles. And when we have germ-free fish, what we end up finding is the fish are relatively hyperactive compared to the conventionally reared fish. They move more. This is a similar finding to that which was observed in germ-free mice in behavioral assays. The germ-free mice are more anxious, they explore more than their conventionally reared counterparts. And what happens when we add 10 micromolars of benzoapyrene? we no longer see a difference in the behavioral activity of these two different groups of fish. The germ-free fish become relatively hypoactive. They decrease their activity, where the microbiome colonized fish become relatively hyperactive. That hyperactivity is what I mentioned at the very beginning when I said benzoate pyrene is increasingly being found to induce excitability in vertebrates. But in germ-free fish, we don't see that trend. The microbiome appears to be mediating how the fish is experiencing benzoate pyrene and impacting its behavioral response. All right. So this was just one foray on one chemical where we varied one parameter of its exposure. There's a tremendous amount of work to do in exposure science and understanding how different kinds of exposures or combinations of exposures impact the microbiome. But ultimately, we're optimistic that we can use zebrafish as a high throughput screening system to zero in on exposures that perturb microbiomes to elicit physiological impacts, and then use that information to prioritize subsequent work in mouse models or other more translationally relevant research systems. So the last vignette that I want to briefly point you to today is some of the work that we're doing to zero in on whether or not the microbiome's linked to our evolutionary history. And I have to admit, this isn't an idea that I walked into on day one of the job and said, that's the problem I'm going to solve. I read some great work from some other people who've been investigating this problem for some time, including your very own Microbiome Center Director, Seth Bordenstein, who had collectively uncovered a trend known as phylosymbiosis. And if you're not familiar with it, phylosymbiosis is a pattern that emerges when the evolutionary history or the phylogeny of a variety of different host species correlates with the similarity in the respective microbiomes of those sequences, of those same species. It suggests that there's some sort of correlated process that is driving host evolution and impacting microbiome diversification across its hosts. Could be something selective, not necessarily something selective in terms of the role that evolution may be playing in producing this particular pattern. Well, we decided to look at some non-human primates and compare and determine whether this pattern of phylosymbiosis, which had been observed in a variety of other study systems at the time, but not explicitly evaluated across mammalia, we decided to determine whether or not, using publicly available data, we could observe similar patterns of phylosymbiosis in mammals and especially within non-human primates. So we innovated an approach that I'm not going to take the time to introduce today, but it's an ecophylogenetic approach that resolves monophyletic clades of gut microbes that manifest particular statistical patterns across sequence. And we used it on some data that Howard Ochman's group generated a long time ago at this point, where they had a variety of different non-human primate communities or populations that were sampled, as well as Western individuals or individuals in the United States, as well as what they called non-Western individuals, individuals from Malawi and Venezuela. And what we ended up finding is that there were very strong species-specific effects in the composition of the microbiome. And in particular, when we looked at the phylogeny of the hosts, as well as, the inferred phylogeny of the hosts, as well as the composition of the microbiomes, we found this pattern of phylosymbiosis was emerging yet again in the non-human primates. Well, what we were particularly interested in doing is, since we were looking at these clades of bacteria and their distribution across these different hosts, we were very interested in determining how the Western individuals that were presumably subject to the process of industrialization, more so than, say, other individuals, how their microbiomes may have, we might have inferred them to change over time relative to these other lineages. And what you see on this, this dendrogram is the evolutionary phylogeny of these different hosts. The number above the lineage indicates the number of microbial clades that appear to have been gained in a subtending group of hosts. The number on the bottom corresponds to the number of microbial clades that appear to have been lost from a subtending group of hosts relative to the other groups that all carry that clade. And an interesting pattern emerged when we looked at this data. There was a very strong, statistically significant enrichment in the number of clades that individuals living in the United States, presumably subject to industrialization, there was a number of clades that they appeared to be missing that all other individuals that we considered in this analysis carried in their guts. Okay, so this suggests that there's something that has happened with respect to life in America, whether it's geography or if it's something that we are doing on a more consistent cultural basis, that's selecting against our ability to access clades that are otherwise represented in our siblings and our cousins. So to zero in on maybe some more information about what could be going on here, we've recently started another type of investigation where we've applied metagenomic sequencing to a similar sort of a framework. Here we generated shotgun metagenomes from a variety of animals, and it's important to note that these all came out of facilities. These are not wild animals that we're looking at here, which is a huge caveat when we think about evolutionary considerations. I can explain if you want to hear some more argumentation about why we think it's still meaningful. Well, we've collected metagenomes or generated metagenomes from a variety of different macaques, humans, mouse, rats, cows, pigs, and dogs, all right? And we asked, how does the functional capacity of these metagenomes vary across these different host species? And if you just look at the raw data, that's what you generally see. There is some evidence of species-specific clustering, and there is some evidence of correlation with the phylogenetic distance across the hosts, as shown here in this particular correlation plot. The x-axis is the phylogenetic distance between the hosts. The y-axis is the Bray-Curtis dissimilarity of the kebrorthology group profiles for each of those different hosts. And you can see here that there's a significant monotonic increasing relationship here between evolutionary distance and functional beta diversity. Am I convinced that this is a form of functional phylosymbiosis? No, not really. I think we have a lot more work to do here to look at other individuals, greater sampling of phylogenetic distance, just to see how this space gets fleshed out. We recently tried this by grabbing some additional publicly available data. This is from a paper quite a while ago, but the nice thing about it is it had very shallowly sequenced metagenomes from a large number of mammalian lineages. And we still see a significant Spearman's correlation, monotonic trend, in these data, but it's awfully variable. Again, what we kind of think is going on is there are a lot of housekeeping genes that are obscuring information about correlations between evolutionary history and functional capacity. So what we're currently working on is to zero in on specific protein families that may explain variation across the tree of life with a higher than expected chance. And that's an ongoing investigation in the shop. But regardless, at the end of the day, there's still a lot of work to do to understand how the microbiome links to mammalian evolution. But we and others in the field seem to be coming to the conclusion that at least in some study systems, including humans, there is some evidence of a meaningful connection between our ancestry and our microbiomes, and that we may be perturbing that ancestral relationship with certain aspects of how we're living today. So let's synthesize all of these different ideas for just a minute that we've discussed today. First and foremost, I think the research that's been done so far, not just by us again, but by many in the field, and there's a rapidly blossoming scope of research that tends to support the hypothesis that we're partaking in activities that are perturbing our microbiome's ancestral connection with our physiology, and that may then mount or affect chronic disease incidence or severity. Have we shown cause and effect? No. No. Will we by the time I'm done with my career? I have no idea. This is a long, long road. But it's a transformative concept because it forces us, or at least encourages us, to reevaluate how we approach medicine, particularly for these diseases that are notoriously difficult to diagnose. Hard to prevent, difficult to manage. It's certainly expensive to manage. And for many of them, they have no therapeutic. So if we can make inroads here as a community and continue to advance the narrative and determine if this hypothesis holds water, we may have a transformative effect on a major biomedical problem. And so at the end of the day, it's really incumbent upon all of us who are interested in this problem to work together, to share knowledge, to determine objectively, does this hypothesis hold water? Does it have merit? And should we continue to advance it forward? Ultimately, we've really got to move towards cause and effect. It's not enough to just show these statistical connections. We have to show that the changes to the microbiome that occur from exposure are perturbing physiology, and particularly the perturbing physiology because of that ancestral connection. So one way we can do that is we can integrate data across these different narratives that I've introduced to you today. Are the protein families that connect to ancestry, also the protein families that are sensitive to exposures, and are they also the protein families that link to disease? That gives us a really strong hypothesis that we should follow up on. But we also need experimental tools to chase cause and effect. And I think this is the biggest limitation we face today. And then the last thing that I want to note is that I'm not here today to argue that the microbiome is a silver bullet. I don't think that it is. In fact, I think of the microbiome as just a component, a variable in a multivariate equation of physiology and health. What we are trying to do is determine the loading on that variable. How much does it matter to health? But we cannot ignore the other covariates in that function and their interaction. And one thing that we could probably do better as a community is ensure that when we're conducting studies, that we're releasing information, particularly about metadata or covariates of the individuals we're studying, that can inform on these types of integrations with more effectiveness in information content than we currently are. There's a lot of challenge there administratively and bureaucratically, but I think we can get there, and that will have a big effect on how we can better understand health as a whole and the microbiome's role in contributing to it. I have a lot of people that I'm very grateful to for the research that I've described here today. I have a very extensive group of collaborators who have all helped advance the various projects that I've discussed. There are a couple of trainees that have come out of the lab. Courtney Armour was a PhD student that led the metagenomics work. She's now in the Schloss lab at Michigan. Holly Arnold is a vet student that contributed to some of the phylosymbiosis work, and she's now a PhD DVM working at OSU. Chris Golke is a very talented assistant professor who came out of our lab, who's over at Illinois, and he led a lot of the work in zebrafish. Y'all should have him out for a talk. He's brilliant. Kristen Cashaw runs our core facility and is responsible for generating basically every single 16S library that we work with. And then I have a couple of current trainees, Keaton Stagaman being the one who has really advanced all the benzoate pyrene work in collaboration with another PhD student, Michael Seeler. We couldn't do any of this work without the support of our funding agencies, especially the NIH. And lately the NIEHS, who's made a lot of the transformations in technology and experimental infrastructure on the zebrafish system possible. And I'm indebted to my colleagues at Oregon State University, especially the other investigators in the Oregon State University Microbiome Initiative. They create an enriching and community supportive spirit around the work that we do, and they afford us all sorts of unique opportunities to ask different questions. I'm sure it's very similar here at PSU, and I know you all really value that type of culture amongst your colleagues. So thank you very much again for the invitation and for your attention.
Thank you so much for an amazing talk. We'd like to encourage the first question to come from trainees. So who wants to ask the first question?